We haven't talked about special senses. The senses we've talked about so far have been general senses, right? These are ones that have a general sensation associated with them, either touch or pressure. You know, they're kind of non-discriminative in terms of what type of stimulus sets them off, right? It's just a touch is a touch is a touch. It doesn't matter what type of touch it is or who's touching you or what thing is touching you. Whereas our special senses, here we're talking about specific stimuli and specific organs that are going to be triggered by these stimuli. So here we're talking about smell, which we call olfaction, taste, which we call gustation, vision, hearing, and equilibrium, or vestibular sensation. Those are our general senses. So these all have special sensory receptors associated with them, and some of them have cranial nerves associated with them. So the cranial nerves and spinal nerves are associated with these special sense organs, and so we're going to go through each sense, kind of break it down, how it works, what the receptor is, what the stimulus is, how it's detected, the pathway, and all of those things for each individual sense. We have to review this concept of transduction. Transduction is this kind of relay of the information. So somehow the information has to go from, we'll just use the eye as an example. Somehow it has to go from a light impulse into our eye to a nerve impulse going back to our brain, right? Think about our sense of smell. Somehow the molecules in the air have to go from that to actually an impulse in our brain. And that's what transduction is all about. It's kind of about translating the signal, right? Getting the signal from whatever the molecule or stimulus is into a nerve impulse so that we can see it and understand it and associate it with things that we might recognize or things that we should be afraid of or things that we should respond to, right? So the stimuli are going to be, you know, the actual chemicals or the light or the sound waves. The stimuli are going to be what triggers the receptor to activate. And so in general senses, again, pain, temperature, whatever, it's all being sensed by those free nerve endings, right? And those free nerve endings kind of come up through spinal cord, you know, through the different column pathways and go to the different areas of the brain through the thalamus. Special senses here, these are going to go through special sensory receptors, special sensory neurons going to special areas of the brain. So remember before we talked about the thalamus being our relay center for all of our sensations. You know, something touches you, yep, it goes up through your spinal cord, goes to the thalamus, and then it goes to your cerebral cortex. Well, our special senses, yeah, things are going to be relayed through our thalamus, but the thalamus isn't our key player here. The association areas on our cerebral cortex are the key players when it comes to our special senses. It's really hard for you to have fragrances or scents going into your nose without you being aware of it, right? You're gonna be aware of it if something smells good or something smells bad. So the thalamus is not quite as integral at kind of deciding what it is we're aware of and not aware of. It's hard to not see something if your eyes are open. You're gonna see it. Your thalamus doesn't need to decide what you see and what you don't see, right? So that's how it was in our general senses, right? You're not thinking about how your socks feel on your feet until I bring it to your awareness, and then you're thinking about how your socks feel on your feet, right? The thalamus doesn't play that same role here. So it's set up a little different. So let's talk about olfaction to get started. Olfaction is our sense of smell. So here we're talking about odorants or chemicals that are dissolved in the air. That's what an odorant is. An odorant is a chemical that's dissolved in the air. It comes into our nasal cavity, we inhale it, and it dissolves in the mucus of our nasal cavity. And then also within the mucus of our nasal cavity are little fine extensions of our olfactory nerve. They're little hairs that are in there. And those hairs detect the chemicals, and that's how we smell. So olfactory neurons, these olfactory cells, are a type of bipolar neuron. Remember what a bipolar one is? It's where the cell body's kind of in the middle there. There's also basal cells. So basal cells, you might remember the stratum basal of our integumentary system, a little bit of final exam review for you. The stratum basal, which is that bottom layer of our epidermis, is where new skin cells come from. Well, basal cells here do the same thing. They regenerate those olfactory receptors. So our olfactory receptors can actually be replaced and renewed. as a general rule of thumb, right, neurons in our central nervous system can't undergo mitosis. They can't be replaced. You're born with all you have, and then slowly throughout your life they die. In our peripheral nervous system, which is what these are, they can regenerate, and they can do that through these basal cells. There's also some supporting cells in our nasal cavity. They're there to kind of protect and surround and make sure those olfactory neurons have what they need in order to survive. Our olfactory neurons are what we call chemoreceptors. So we talked briefly about the different types of receptors in our body before. We talked about nociceptors and thermoreceptors and mechanoreceptors. Well, chemoreceptors are special receptors that respond to chemicals, right? So they respond to chemicals. And so we'll talk about these, the olfactory nerve, right, cranial nerve number 1. That's going to be activated by these odorants. That signal travels to the olfactory bulb, back through the olfactory tract. The olfactory tract is going to go and kind of split off to different areas of our brain. Parts of our limbic system. Remember, our limbic system is where memory is. Our limbic system is where emotions are. This is why when something smells good, you're probably going to smile when you think about it, or you're going to be a little happier when things smell good, or when things are stinky, you're like, oh, get me out of here, this place stinks, right? I know that when we were searching for houses last spring, we would go into all these different houses, and if the house had like a smell to it, there was the Chinese food house, there was the cat pee house, there was the mothball house. Like, you remember the smells of those houses or places, right? So it's all linked through that, and we'll talk a little bit about that as we go. So this is the general setup of the olfactory epithelium. You have those little olfactory receptors with their little hairs dipping down into the nasal cavity. Those hairs are, of course, trapped in mucus. That's why our nose is always a little moist, right? That mucus, if you remember from last night, that mucus is saturated from that nasolacrimal duct, right, draining our tears. That's what keeps our mucus nice and moist in our nose. And so those odorants come in, they dissolve, and then the chemoreceptors there detect the chemicals and send the signal up and out to the brain. So they get activated by these odorant binding proteins, right? These special molecules that detect the odorants. They kind of capture them. You know, they're literally sitting there waiting to grab any molecules, any odorants that are kind of wiping past or, wisping by. And so this is our pathway here. We have, forgot to get my colored markers out. There's only black here today. Grab some new ones. Okay. So we have odorants. Odorants dissolve in the nasal cavity. And then they're going to bind to a receptor, an olfactory receptor. And when they do that, they activate a G protein. We talked about G proteins briefly when we did the endocrine system. G proteins are what we call a second messenger. When something binds to the outside of a cell, a G protein is like, oh, okay, I'll go get my mom. Right? That's like ringing the doorbell and then the little kid answering the door. That's what the G protein does. The G protein takes that signal from the outside and makes the inside of the cell do something. The odorant itself doesn't actually come into the cell. Right? And so what it does is it's going to trigger adenylate cyclase to basically turn into this thing called CAMP, or cyclic AMP. And what cyclic AMP does is it's going to open sodium channels and allow a depolarization. The cells depolarize, the action potential is sent. So, I'm going to abbreviate, AP is sent. So then you might wonder, why don't we just have chemically gated channels on our olfactory receptors? Remember, a chemically gated channel is a channel that's going to respond or open when a chemical, such as acetylcholine, binds, right? The reason is it's kind of a backup plan. It's a cross-checker. It's kind of like a safety on a gun. You want to make sure that this is, in fact, something that's supposed to send an action potential. Because our special senses, taste, smell, vision, equilibrium, hearing, these special senses are so fine-tuned and so important to our survival and to our homeostasis that when signals are sent, they need to be true, right? We can't have just anything sending off an action potential. So these odorants dissolve, and they're not just binding and causing a depolarization. They bind, they cause a chemical change in the cell. This is the cell kind of checking and saying, okay, are we sure that the person that's at the door is the right person? Are we sure they're really who they say they are? Okay, cool. Now we'll send an action potential. It's a backup plan. It's a way to double check. And so that's what we call transduction, right? Transduction is this process. This is olfactory transduction, if we want to give this a label. So the pathway then, we take those neurons and they're going to synapse with other special cells, and eventually they're going to travel down our olfactory tract. Right, the olfactory tract, you can see that on the brain model in lab, right, that olfactory tract. It's going to go to the primary olfactory cortex, which is located on the temporal lobe. This sense actually does not go to the thalamus at all. There's no relay. Olfaction is one of our primary survival sense. It's a built-in evolutionary mechanism that basically says if you smell fire, if you smell a dangerous chemical, you need to be alerted immediately. And so there's none of this synaptic delay that comes from relaying the signal through the thalamus. Olfaction goes directly to the brain. So with all of our other general senses, we'll have a pathway through the thalamus. Olfaction, not so much. So our primary olfactory cortex is where all this magic happens. From there, the fibers kind of radiate out and they go to different areas. So from the primary olfactory cortex, it's going to go to parts of the limbic system, right? Which limbic system includes all of these structures listed here. The amygdala, the hippocampus, right? That's all part of the limbic system. When you think of limbic system, you should think of emotions and memory. Parts of our hypothalamus, right? When you think of hypothalamus, you should think of hormones and homeostasis. Right? This is why when you smell food, what starts happening? You might start salivating, you might say, oh man, I'm hungry. It's been a couple hours since I've ate something. That's your hypothalamus doing that. Right? Your hypothalamus keeps you in homeostasis. You smell food, and now you're reminded, oh yeah, I need to eat. My blood sugar's kind of low. Right? So this is why our olfactory system gives us such an emotional, immediate response, right? Have you ever smelled something and then it reminds you of something from your past, right? I know whenever I walk through a department store, which I rarely go to the mall anymore, I don't think many people go to the mall anymore, but I know when I go to the mall and I walk by the cologne counter, it reminds me of middle school because I felt like every dude in middle school had a ridiculous amount of cologne on. No offense, guys, but it reminds me of the little boy. I mean, I didn't date a little boy. I was a little girl. The kid that I dated when I was in middle school, his name was Ricky. I remember Ricky, and I'm like, oh, I remember we would stand there and we would talk. Oh, he was so nice. Like I'm literally taken back when I walk by the cologne counter. Like it takes me right there. And I'm sure you guys all have things like that. Like there's a fragrance or there's a thing that you smell that reminds you of your grandmother or reminds you of somebody you used to have a relationship with or something that you didn't enjoy that gives you a bad memory, right? It's all because of that limbic system. So that's olfaction. One and done. We got 5, we're down 1. We got 4 more to go. Gustation. It's our sense of taste. So this is very similar to our sense of smell. In fact, almost 70% of what you taste is dictated by what you smell, right? Which is why that old trick of like plugging your nose and eating it anyway, it does work. It does work. If you plug your nose and eat something that you don't like, you will taste it less. The problem is your mind already identifies the thing you're eating as something gross. So you probably still think that it tastes gross, even though you, in fact, can't taste it at all. So this is the same concept. We have chemoreceptors and we have chemicals dissolved in something, except here, instead of the chemicals being dissolved in mucus, they're dissolved in our saliva, right? That's what is in our mouth. So we're actually combining olfactory reception and gustation. There's kind of two factors here that come into play when we're tasting something. So our taste buds have to get stimulated. And just like our olfactory receptors getting stimulated, our taste buds need to detect dissolved chemicals. And so the dissolved chemicals come into our mouth, we ingest them, and then they're going to bind to our taste buds. And then, just like before, that's going to cause a depolarization, which will send a signal back to our brain and allow us to understand exactly what it is we're tasting, right? Tasting different chemicals. Each taste bud in our mouth is associated with a single sensory neuron. It's going to carry that information back. So let's talk about our different taste buds. They're all a little different, and you know this if you've ever looked at the surface of your tongue, which you have. You might not want to admit that you've stared at your tongue in the mirror before, but I'm sure you have. Right? There's some big lumpy ones in the back that look kind of gross, right? The majority of the tongue surface looks kind of normal. Have you ever looked at a cat's tongue? Right? The front part of a cat's tongue looks like a comb almost, right? We have those same markings on our tongue, they're just not as big. So our taste buds live in these little mounds on our tongue that are called papillae. So papillae are what we think of as taste buds. So we look at our tongue and we see all these big circles and lumps and we call them taste buds. But really, taste buds are inside those mounds. The mounds themselves are papillae. So we have what's called valate or circumvallate papillae. These kind of cover the back part of our tongue and the sides of our tongue. This is, in fact, the majority of our taste buds. The majority of the papillae. They contain hundreds of taste buds. So each one of those mounds contains hundreds of taste buds. So the taste buds are the actual receptors. Right? They live in the mounds that are on our tongue. We have fungiform. Fungiform are in the very back. They're very big and lumpy. When you go, you can see them in the very back. They look kind of gross. Those are the fungiform. They contain very few. And then the other kind that's not listed here are called filiform. Those are the ones that are elongated on cats. They don't really have any taste function, but they're there for kind of grasping and swooping food into your mouth. So think of like licking an ice cream. Have you ever licked ice cream and then looked at the ice cream and there's lines on it from your tongue? That's gross to think about. But those lines are from the filiform papillae that are on the front of your tongue. It's the same type that are on cats. The cats are just much longer. Oh, there they are, filiform. They were in the next slide. So the filiform don't really contain much taste buds at all. They are more for texture, detecting the temperature, texture of food, bringing it into your mouth. So all of those taste buds are going to be linked to either cranial nerve 7, facial nerve, right? Cranial nerve 9, hypoglossal nerve, or cranial nerve 10, vagus nerve, right? All of those cranial nerves are associated with taste. There's basal cells here also. So just like in our olfactory sense, we had lots of replacement going on. You have replacement here, and I didn't mention this before, that rate of replacement slows as you age, right? So if you've ever watched an older person, I'm talking like in their 70s and 80s, like an older person eat, sometimes they'll put really crazy flavor combinations on their food. They'll use like a lot of hot sauce or horseradish or something to get some flavor to it because their taste buds aren't working anymore. They have to kind of fire it up in order to actually taste something. Same thing with sense of smell, right? Older people sometimes lose their sense of smell. They can't smell the fact that their house smells like pee or whatever, right? They can't smell it anymore because those basal cells just aren't replacing the worn out cells. And there's supporting cells in the taste buds as well. They're there just to nourish, protect, and support those taste buds, make sure they have what they need. So here's the structure, right? We have all of those papillae on the surface of the tongue. but the taste buds themselves are living in those mounds. Those mounds have lots and lots of little taste receptors, or taste buds, taste bud, taste receptor, same thing, right? Those chemicals that we bring in, we chew them up, we break them down, they dissolve chemically in our saliva, and then they go into those little pockets, they go into those little papillae, and then they're detected by the taste buds. And so here you see the kind of the villi, those little extensions, kind of like the hairs on our olfactory receptors, kind of sweeping around and trying to taste the chemicals that are being dissolved. So our taste sensations, we have 4 main taste sensations. We have the sensation for sweet, right? That's why some people, we say, have a sweet tooth. It's literally just detecting glucose or fructose in the food that you eat. There's sour taste. Sour tastes are actually activated by acid. Hydrogen ions will activate those sour taste buds. And so within these clusters of papillae are special taste receptors for sweet and sours, right? So they're designed to detect, right? Whereas in olfaction, an odorant is an odorant, right? It's just going to bind, and then it's going to send that chemical signal. Here we have different types of taste buds that are going to react and respond to different taste chemicals. We also have salty, right? Salty tastes are obviously going to be activated by ions like sodium, right? Bitter flavors are going to be activated by nitrogen-containing compounds. Usually these bitter things are things that are poisonous, so this is a defensive mechanism to be able to taste that bitterness in food. And then there's also something called umami. It's another variation of flavor, and this is it's kind of a savory flavor, like Parmesan cheese, or like a broth, like beef broth is kind of what it tastes like. So this is that kind of meat and cheese type flavor, strong cheese. Any type of protein really is going to fire up those umami taste buds. So our taste receptors get activated. We'll kind of break this down. Call this, whoops, that pen's not going to work. We'll call this the gustation pathway. So our taste receptors will only detect one of those flavors, right? One of those variations, but they'll become activated. In order to be activated, that chemical has to dissolve in the saliva. So chemicals. So here's the tricky thing, right? So saliva is water, right? It's water-based. So what about oily substances, right? Oil doesn't dissolve in water, right? So how do oily things get tasted and detected, right? If there's a poison that's hydrophobic, if there's a poison that's oil-based, how do we detect it, right? That's why some substances are really, really dangerous. And substances you can put into your body and not even know that it's poisonous until it gets down into your digestive system. So this includes some drugs and things like that you would put into your body maybe accidentally, because they can't dissolve in the saliva. They need to be broken down more in order to be detected. So we're going to depolarize by opening calcium channels here. So calcium ions are going to enter the cell. So we have depolarization. Chemicals dissolve in the saliva. This is going to activate calcium channels. Why wouldn't we activate sodium channels? Every other time we've always talked about activating sodium channels. Sodium rushes in, blah, blah, blah. Why would we activate calcium channels?
Because we need muscles to work.
That's a good idea. Maybe because we need the sodium for our muscles. What's in our food? We're bringing in sodium, right? So if we're bringing in sodium, how are we going to detect it if automatically all of our sodium channels open, right? So there has to be a slightly different mechanism here in gustation, because we're bringing in sodium by our diet, right? By the food that we put in. So calcium channels open. Calcium enters in. And that will produce an action potential. AP triggered. It's a slightly different transduction mechanism because of the fact that we're talking about food, and a lot of our food contains sodium, and our salty taste receptors are going to be triggered by sodium. So those ions come in, they dissolve, trigger an action potential. Oops. That sensory neuron, so the taste receptor itself is going to respond by calcium, but the action potential that's triggered in the sensory neuron is going to work off of sodium. AP triggered in sensory neuron. And this is a normal action potential that we're used to thinking about. So I'm calling it a quote, normal action potential. So this one's going to respond to sodium and do that whole thing. This piece here is just the taste receptor, aka taste bud, that's going to do that. That's going to open calcium channels. Okay, so here we're talking the facial, hypoglossal, vagus nerves, right? Those are all going to go to parts of our medulla. Let's think about this. So trigger the sensory neuron. This is going to go to medulla. It's something called the solitary nucleus. Why would it go there? What does our medulla regulate? Heart rate, blood pressure, right? But our medulla oblongata also regulates our digestive system. It plays a huge role in our digestive system. So if we're putting something into our mouth, the rest of our digestive system needs to know about it. So our medulla oblongata, that's gonna, I wrote medullary, sorry, medulla. Medulla oblongata, the solitary nucleus, that's gonna activate it, and then our medulla can tell the rest of our digestive system, hey, we got something coming. We just put something in our mouth, you need to prepare, we need to start secreting enzymes, we need to get churning, etc. That information is then going to also go to the thalamus, right? So now we're going through that familiar pathway. So solitary nucleus is going to send information to digestive system, and the thalamus. Right? The thalamus will then send that information to our primary gustatory cortex. Hey, that tastes good. Hey, that tastes bad. That's where we're then aware of what it is we've put in our mouth, the texture, the flavor, the temperature, all of those things. And we can make an informed decision as to whether we want to keep eating it or not. Right? Some of this information also goes to the limbic system. Right? So from the thalamus, it'll go to the primary gustatory cortex and limbic system. Why? Well, no one really knows why, but it goes there, and that's why sometimes we eat to feel good, or sometimes we say, man, I've had a bad day. Can we just go get pizza? I just want to eat pizza. I don't even care, right? Or I really need cheesesteak, or I need cookies, or I want to eat a gallon of ice cream, or whatever those things are that give you good feelings from eating. You can thank your limbic system for that, right? Because our gustatory pathways run through there. It also gives us an emotional reaction to taste. Not only is it going to bring about some emotional eating, but it's also going to trigger that, oh, I love my mom's lasagna. Oh my gosh, I can't wait to eat it. And then you start smiling when you start thinking about your mom's lasagna, and you just, you can imagine eating it, and your mouth starts watering just thinking about it, and all of those things, right? So the limbic system gives you all the good feels about whatever it is you are eating or whatever it is you think you're going to eat. And vice versa, right? It makes you say, oh, no, I'm not going to eat anything that woman cooks because it is horrible, right? So let's go to our eye. Olfaction and gustation. I never like to use the word easy because nothing's really easy in A&P, but of all the five senses, those two are the easiest. They're the simplest, they're the easiest to kind of break down and understand. the eye, we're gonna get a little bit more complicated. There's a lot more to it. We looked at the anatomy of the eye last night, so hopefully some of that knowledge will help you a little bit understand some of the things that we have going on here. So our eyes are packed in, I just realized we did not dissect the eye last night. No one called me out on it, but if you want to dissect an eye, we can always do it next week. I just realized we didn't do it. But the eyes are packed in fat. So they're in there, right? They're protected by bone, but kind of behind our eyelids are lots and lots of fat, which is why super duper super anorexically thin people and or people with hyperthyroidism where they're having trouble retaining body fat, their eyes are very sunken in because the fat that's there isn't there anymore, right? There's lots of fat surrounding all of our bony orbits. Our eyelids, which are also called palpebrae, right? Those are protecting the eye. They prevent any foreign objects, debris, things like that from going in. They also help to distribute those tears, wash the tears over the surface of the eye. We drew that little pathway last night also. So there's a couple other glands in there. There's something called a tarsal gland. These tarsal glands are thick, kind of oil type glands that are associated with our eyelashes, right? Have you ever looked carefully? Probably most of the girls in here have, unless anyone else in here wears eye makeup, but when you look really, really close to your eyelids, right, at where your eyelashes are, you can see it almost looks like sweaty in there all the time, right? Because there's special oil glands associated with each and every eyelash. That's why the eyelashes have a different texture than any other hair on your body, right? They're kind of thick and they tend to be differently pigmented because of that. So the tarsal glands, they used to be called meibomian glands, but then they changed the name. That's what those are. So the canthi are just the little corners. So we have a medial and lateral canthus. That's just the corners of the eye. The caruncle, we talked about that last night. The caruncle, my uncle drives a pink car. That's a little pink triangle in there. And then orbicularis oculi is the actual eye muscle. We've talked about that one before. Mine keeps twitching. My right eye has twitched for about 30 weeks now. And I keep Googling, like, why would my right eye twitch? And it's either stress or there's like a spiritual definition that says, someone's talking good about me. Highly unlikely, but I'll go with it. So anyways, there's our canthi, the corners, caruncles, the little triangle there, and then the palpebrae, or the eyelids themselves. Oops. I think it's going to start to freak out. All right, hopefully it doesn't. Eyebrows and eyelashes are there for protective measures. Our eyebrows are there to really prevent any forehead sweat or head sweat from coming into the eyes. Any dust or debris or anything that happens above the eye, the eyebrows are there to catch it. That's why there's that extra ridge, that superior orbital margin that's there. And then the eyelashes, of course, are there to catch things as well. That's why we reflexively blink when we see things coming at our face to try to prevent those eyes. The conjunctiva is the inner lining of our eyelid. There's actually an extra piece of it called the ocular conjunctiva that covers the white part of our eyeball also. In lab, when we identified it on the models, we just said that it was kind of that pink inner eyelid structure, because on the model, we either have the cornea or the sclera on the eyeball. There's no extra layer of conjunctiva, but there is in real life, and you know this if you've ever had conjunctivitis or pink eye, that your eyes are very pink, very inflamed, very itchy, irritated. And it's because the inside part of your eyelid is inflamed, as well as the surface of your eye, that extra ocular conjunctiva. So the lacrimal gland and lacrimal apparatus, we mentioned this last night. Also, it's lateral, on the lateral side of our eyes. It's releasing tears as well as a little bit of mucus, right, to help sweep and lubricate the eye. It's stimulated by the autonomic nervous system, which is why sometimes when you're in that fight or flight mode, you might cry, right? Oh my God, I'm so scared right now, right? And you might cry. It's reflexive, and different people have different responses. Crying is actually one of those things that no one really understands why it happens. There's lots of research about why it happens. There's other animals in the mammalian kingdom that they They cry also, but no one's really pinpointed exactly what the trigger is because some people cry more, some people cry less, and they don't really know. But the lacrimal gland is there to wash away debris, to lubricate, because there's no blood flow to that part of the eye, so there's no way to get nutrients there. So your tears are what's not only washing the eye, but also bringing nutrients in. Think of tears as kind of like the cerebral spinal fluid for the outside surface of the eye. Right? There's no blood going there, so we have to have some way of getting nutrients. So that's what our tears do. And of course they drain into the nasal cavity. That lacrimal sac catches it and it drains through the nasolacrimal duct into the nasal cavity, so it helps moisten the mucus in your nasal cavity. The puncta is the hole that kind of drains all of that. So there's our structures, lacrimal gland sweeping across through the puncta, down through the nasolacrimal duct, and into the nasal cavity. This is also why when you cry a lot, you get that ugly cry going, and it's called the ugly cry because you start like snotting and dripping out your nose, but that's why, because it literally drips out your nose. That's what it's designed to do. So the eye muscles, we labeled these last night as well. They're paired, it's pretty easy to follow along. Superior and inferior rectus, medial and lateral rectus, and then our superior and inferior oblique muscles. Each rectus muscle moves the eye in a direction. The reason why these are so important is because think about how they move, especially the medial and lateral rectus, right? When your eyes look this way, I'll do it this way, I can't turn my wrist that way. When our eyes move that way to look, it's the lateral rectus of this eye that's contracting and the medial rectus of that eye that's contracting, right? Otherwise, you're going cross-eyed if both your medial rectus muscles are contracting. So this is why eye muscle development and kind of that tracing of the finger after being hit in the head is really crucial in diagnosing concussions and brain injuries and developmental delays, because there's so many. I mean, we learned all the cranial nerves. There's like 6 cranial nerves that are associated with the eyeball, right? And so understanding that pupillary light reflex and some of the other reflexes and functionings of our eyeballs, that will help us understand, oh, there's something going on there. That's actually one of the things that I was kind of going towards when I was looking at my eye twitch. I'm like convinced I have a brain tumor right now. I'm like, I teach A&P, but I have hypochondria, which is awful. It's a bad combination. So I told my husband, like, it's been twitching for 3 weeks. I'm pretty sure it's a brain tumor because I've had this headache and blah, blah, blah. But nowhere because Google is the ultimate doctor, right? Nowhere in Google world does it say eye twitching can be related to a brain tumor. So, but I'm convinced it is somehow. Kind of psycho sometimes. Okay. Inferior oblique, superior oblique, we've talked about those too. We've also talked about the cranial nerves. Cranial nerve 4, 6, and 3 are all related directly to eye movement. So let's talk about the eyeball structure itself. We alluded to the structure of it last night, the idea of these 3 layers. We even kind of drew it out. I'm going to draw it again just because repetition helps with memorization, right? Learning it. And so we kind of have this big old ball, and that's really light. And up here we have, kind of gets a little bigger. So we have 3 major layers to our eye. We have something called the fibrous tunic, or this outer fibrous layer, right? The outer fibrous layer is our sclera and cornea. So we'll do this too. Our sclera and cornea, right? So that's going to be this outermost layer. This is the part that you can see. So it's called a tunic or a layer. So a tunic is just, it's another word for a layer. I always learned it as a tunic. So, but fibrous layer, fibrous tunic, same difference. The fibrous layer is made of that sclera and cornea. We mentioned these. The sclera is that white part of the eye. The cornea is the clear cover, only on the front part of the eye. So if we were to kind of pull your eyelid back and look really far back there, there's no cornea on that part of the eye. The cornea is only present on the part of the eye that we see. The vascular layer is next. And notice I'm only drawing this on this inside part of the eye. I'm not going all the way around. So this is our vascular layer. And then the neural layer is the very innermost piece. It's a squeaky purple. The neural layer. So there's what we just drew. I know you can always just look at the picture, but it's so much better to actually draw it. So let's break it down a little more. We have those three tunics. We have the sclera, or that fibrous tunic. We have our choroid, or our vascular tunic, and then on the inside is where our retina or neural tunic is. We're going to go through all of these structures bit by bit like we did last night. Let's talk about that sclera. So this is mostly made of collagen, so it's a connective tissue, lots of fibers, hence fibrous layer. And this is the white part of our eye. The cornea is translucent, or the cornea is clear. Right, remember that. And it's avascular, right? So there's no blood vessels there. This is allowing the light to pass through. This is why the sclera stops and the cornea starts, right? So the cornea is going to cover the eye and allow light to pass through. So looking a little closer, we see the sclera, and in this image you can really tell that it's lots and lots of fibers, connective tissue fibers, whereas that cornea is just a thin film. So we get to the vascular layer, which in general is called the choroid. We have the choroid. This is all of our blood vessels that surround the eye. I'm abbreviating BV. And the ciliary body, I know I'm going to write a lot over there, so I'm trying to conserve space. The ciliary body, which the ciliary body is where our iris kind of comes off of. Yeah, iris, which is the muscle. And the suspensory ligaments. For our lens, all coming off of that ciliary body. So the suspensory ligaments, the iris, the pupil, all associated in this vascular layer. The suspensory ligaments are those little kind of like puppet strings attaching to the lens. We'll talk about the lens in just a minute, but the whole purpose of the lens is to fatten or flatten, to refocus the light on the back of the retina. And the iris is the colored region that lets the light in. This is going to determine the amount of light that's coming into our eye. So there's melanin and other pigments in your iris. That's what gives your iris the color. Blue eyes just lack the pigment. So that's just a closer look. You can see the ciliary body there. Suspensory ligaments are the little puppet strings holding our lens in place. So there's a couple muscles associated with the iris. So our iris has what's called a papillary sphincter and a dilator muscle. And we talked about this a little bit last night. I spelled dilator wrong, dilator. So the dilator muscle is going to increase the pupil size. The sphincter is going to decrease the pupil size. So the sphincter muscle is under parasympathetic activation. Our dilator muscle is under sympathetic activation. I'm just abbreviating para and symp because I'm at the bottom and I don't have enough space, but our dilator muscles under sympathetic activation, right? Dilator means get bigger. When you're under fight or flight, when you're nervous for your life, right? When you're walking down that dark alley or walking in your basement, your pupils are going to get bigger to let as much light in as you can so that you can make all the observations and make all the educated decisions that you can. So your pupils are going to get wider. That's the dilator muscle. You're resting and digesting, you're Netflix and chilling, your pupil's going to get a lot smaller so that, you know, because you're not in survival mode then. Your gut's churning, your reproductive system's going, all that stuff's going, but your fight or flight isn't at that point. This is just showing you that concept of getting bigger or smaller. We drew this last night, but those two muscles kind of work opposite of each other. You have this sphincter muscle and the dilator muscle. Right, which is why if you look carefully at your eye, which again, I'm pretty sure most of you have at some point in your life, you see that there are two distinct areas of your iris, and generally, they could even be different colors, right? The inside might be yellowish and the outside might be greenish, or the inside might be brownish and the outside might be bluish, or vice versa, right? Because they're literally different muscles. They have different cells in them, different pigmentation. So, now we get to the neural layer of our eye. The neural layer has what's called a pigmented epithelium, and then it has photoreceptors. So our neural layer is actually two layers. So here I drew it as purple, but I'm going to make it purple and green. So there is a pigmented layer. And then this one, which is the photoreceptors. So just for artistic sake, I'm going to go ahead and draw in here that ciliary body so that we understand where that is. I didn't want to ignore, whoops, I think I touched that, my bad. Didn't want to ignore this. Some little suspensory ligaments. There's our lens. We drew the same thing last night. And there's our iris. And this hole is the pupil. Sorry, I went a little fast there. I'm trying to fix this, but there we go. Okay. So what I just drew was a ciliary body, which is basically an expanded vascular layer on both the top and bottom. Hanging from that are the suspensory ligaments, right, which suspend and attach to the lens, right? The lens is the puppet, the suspensory ligaments are the puppet strings, and the ciliary body is kind of like the puppeteer that's actually going to pull on the strings and make them pull. And then this green section, this is the iris. Because this is a cross section, it's kind of like we cut your eye in half, this sagittal section, and we're looking into the eye. So I just made this person have green eyes, right, because I made it green. That's the colored iris. And then the pupil is the hole in the iris. So that's going to allow the light through. So light, when it comes into your eye, is going to pass through the cornea, through the pupil, and through the lens. So that deep layer, those photoreceptor cells, going back to this neural layer, those photoreceptor cells form the optic nerve, right? Take a look at what I did here. All of these purple and green lines, they're going to come back here, and those bundles, those axon bundles, make up the optic nerve. Yes?
So does the photoreceptors go all the way through with the...
Yeah.
Okay.
Yeah, they're all kind of bundled together at that point. So let's talk about those photoreceptor cells. There's two types of cells. So we have rods and cones, which you've probably learned at some point in your education before. Rods are for kind of basic outline vision. These are the ones that just help you see shapes. These are the ones that help you determine that there's light in the room or no light in the room. So rods are just general vision. So if you like kind of squint your eyes a little bit to the point where everything's blurry, it's just your rods that are activated at that point, right? So if you just have just tiny slits in your eyes, just letting enough light in, that's what it's like to live with just rods, right? When you open your eyes and you can actually see shapes and details and color, that's the cones that allow you to see that. Cones help you with color vision, and they also help you with really fine, distinct vision. So what makes humans so special is our eyes. Our eyes are the keenest in all of the animal kingdom. Our eyes are, our sense of vision, I should say, is our most important sense in terms of survival and in terms of living an adaptable life. So those cones are what make that, because our cones are highly sensitive. So all of those cells come together, bundle up those axons, and form the optic nerve. The ora serrata is that boundary, and you saw this on the models last night. It's kind of feathery looking. It was that boundary between this vascular layer, and I can't really draw it on here because I'm not that good of an artist. But the ora serrata is just surrounding this whole structure here. It's just behind the lens. It's this kind of feathery looking structure, and just the boundary. It's just the transition from non-neural tissue to neural tissue. So on the very back of our retina is something called the macula lutea, or the macula lutea. So this is just kind of to the side of where our optic nerve is. This is our point where most of our cones are. Most of our cones are on this piece right here. The rest of our retina, mostly rods. Cones are right there. The fovea centralis is the center of that. This is our point of finest visual acuity. I talked about this last night a little bit. If you have something in your hand, for example, I'm looking at this marker, I see all the things on this marker, all the details of this marker. I can see all you guys, I can see what you're wearing, but you're all just kind of like a blur, right? I'm not like focusing on you. It's because the light that's going into my eye from this marker is going back to that fovea centralis, and that's how I'm able to see all of the details here. The light from all of you guys is going everywhere else, right? It's going everywhere else. Macular degeneration is a condition where literally that macula is shrinking. In other words, there's less and less cones, right? So people with macular degeneration, they're having less and less field of clear vision. So in other words, in order for them to focus on something, it has to be very close and very big in order for them to actually see it in lots of detail, right? They don't have the ability to look around the room and see the amount of details and colors that we see. So the optic disc is this region right here, because this neural region has all the photoreceptors, rods and cones, except right here, there's none, because this is where they leave the eye. So it's called the optic disc or the blind spot, because there's no photoreceptors there. You can find your blind spot if you kind of close your eyes first so that your brain doesn't really know what's going on. And then you put a pen or a marker or a finger, you can even do a finger, in front. And if you close one eye and you slowly bring it towards you, you will get to a point, and it's tricky because your brain likes to, right about there, your brain likes to fill in the gaps, but you'll get to a point where it disappears. This is a little too thick to do it with. It has to be kind of thin. I used to do this when I taught 8th grade. I'd pick a student, and we'd all make them disappear. You can do it if they're far enough away. You can kind of move your body enough to make anything disappear. The crazy thing is 9 times out of 10, your brain knows what's going on and fills in the gaps. Kind of like how you can't really test your own reflexes, right? Your brain knows what's happening. So it's really hard to test your own blind spot But I think there's a test in your book where there's like a dot and you slowly like move the book away. And you have to only do it with one eye, because if you have it with both eyes, that's how come we don't have blind spots, because our field of vision overlaps to compensate for that. So there is a picture of the retina taken from an ophthalmoscope, right, the little thing that they use to look into your eyeball. And here you can see that blind spot, that optic disc. Oops, that's right there. The optic disc. That's where the light scatters, right? So it's going back there, and there's no pigmented layer there, right? Everywhere else, the light is being absorbed, except there, right? Which is why that spot looks bright. You can always see that darkened area where there's lots and lots of cones. That's that macula lutea, that fovea centralis. Oh, you can also see all the blood vessels, right? So the blood vessels enter that choroid through that optic nerve passageway. So let's talk about the lens. It's held in place by the ciliary body. The lens contains fibers. The fibers are tightly packed together. There's no nuclei in there, so the cells of the lens aren't actively dividing. The lens is translucent, it's sitting there. And just like almost every other part of the eye, there's no blood flow to it. The only part of the eye that has blood flow is the vascular layer. The iris, the ciliary body, right? Those parts have blood flow to it. The rest of the eye doesn't. So there those things are. We see how big that lens is. And if you guys want to stick around next week after the practical, we can chop open an eye. You'll see it's hard on dissected creatures. However, in us, it's like the consistency of about a grape. So it's a little squishy, which is kind of cool. So a cataract is when the lens becomes cloudy. So instead of it being this translucent piece, it gets cloudy. And this is usually because parts of those fibers get kind of tangled up and kind of crystallize a little bit. Trauma, UV exposure, all of that can cause damage to those fibers and can turn your lens color. And you can imagine with that light coming in, that's going to heavily impact your vision, which is why you're not supposed to look directly at the sun or the solar eclipse, things like that, right? Cataracts can't be reversed. You can have cataract surgery, have them removed, but you can't undo the damage of cataracts. You have to usually replace the entire lens. They usually do it with a cadaver lens. I'll just pull one out of a dead body, which sounds kind of creepy, but that's the closest thing to a living, donor, so that's what they can do. So the different cavities of the eye, we have the posterior cavity, which we talked about before. This is the area behind the lens. This whole area here is called the posterior cavity. And it's filled with vitreous humor, which is this kind of jelly-like substance. It's like thick. And it helps keep that light from bouncing around in the eye. It helps keep it directed and focused to where it needs to go. It also helps maintain the eye shape. It kind of provides this intraocular pressure to sort of push open or push out on the walls of the eye so that the eye doesn't like squish in. And then the anterior cavity, which the anterior cavity is then subdivided again. You don't have to know all of that. Just know that this area in front of the lens is our anterior cavity. And this has something called aqueous humor in it. So humor is Latin for fluid. So this is the watery fluid in our body or in our eyeballs. And so the vitreous humor and aqueous humor circulate just like CSF. So it's filtered from these little filtering stations around the choroid, and that's going to allow for this exchange of nutrients and waste products to make sure that our eye has all of the ingredients that it needs. There's something called the scleral venous sinus, or the canal of Schlemm. That's how I learned it. It's just this little tiny blood vessel that drains and filters that aqueous humor, helps circulate it. Glaucoma is a condition where that canal gets blocked. And so all of this fluid can come into the eye, but it can't get out, right? And so what glaucoma does is it's a continual buildup of fluid in here, right? And that pushes on the lens and pushes on the suspensory ligaments and pushes on the ciliary body, and it can damage your eyesight, right? It can detach your lens if the pressure in there is too much, which is why, do you know what a glaucoma test is? Do you know how they do it? Has anyone had one? They like blow like a puff of air on your eye. So they're basically testing, you know, how squishy is your eye. If your eye doesn't squish that much with the puff of air, that tells them that that intraocular pressure is probably too high and you might have glaucoma. If they blow that puff of air on your eye and it doesn't squish, or if it does squish, then they're like, okay, it's probably draining properly, you're good. Glaucoma is, and actually it's an inherited thing. So if you have a family history of it, the chances of you having it are higher. So there's all of the parts of the eye that we went through, anterior, posterior cavity. This line here is a small canal. A small canal that's just allowing for the circulation of that vitreous humor. So it's not on any of our models. There's glaucoma. I just mentioned that. It's the second leading cause of blindness. And it does have a genetic component, but if there's no family history, there we go, no family history, then they don't really know what causes it. They can just kind of up and out. Usually you don't know that you have glaucoma until you start losing your eyesight, so which is why vision checkups are good, even if you don't wear glasses. So I have never worn glasses. I've always had perfect vision until now. Recently I just started to get a little squinty. So I made my very first eye appointment, and I do have a family history of glaucoma, so I'm kind of nervous that I'm like, not only do I have a brain tumor, but now I also have glaucoma. So I'll let you guys know. But anyways, we'll stop here for tonight, and we'll pick up with how we perceive vision tomorrow, or next week.